it's a good thing for the folks who are here. I mean, because I have something to share with you, and you can thank one of the members of your class. And I'm telling all the classes. And I do not know a lot about it. I'll frame it for you. I mean, it's all about artificial intelligence. One of the folks in this class, and I'm sharing with all the students, came to me to discuss the study guide for the next exam and indicated that they had taken that review recording and put it through Chat GPT and said, make an exam. Oh my God. Now remember, I'm reading from the exam effectively when I'm doing that. It's like it was looking over my shoulder. So, I mean, the questions aren't necessarily the same, but all the answers were there. Do this. I have no objection. It's a very controversial area in education. For me, I'll say this, and I've said it, it's all about getting it right. No one will ask you about the events at the neuromuscular junction in your profession. I can guarantee you that. Nobody's going to ask you how many sodiums went in and how many potassiums went out. They don't give a hoot. I was going to say something stronger. Don't look at me like that. All right? But I'm tasked with teaching you this. For most of you, the idea of a difficult science class or any class that's difficult is you have to be able to assimilate, to understand to a reasonable degree, and answer questions successfully to a reasonable degree on difficult material. That's because it only gets harder in whatever field. It doesn't matter what your field of endeavor. It only goes in one direction, hard. Okay. And again, the AI thing. I mean, be careful if you're going to write. Somebody tells you I need to see a paper or a report or this or that. Be careful, because they're assessing other people's intellectual products and putting it to you, and then you will book the fine line. There's a lot of people, a lot of faculty that get very huffy about intellectual property infringement and not doing a proper citation and effectively plagiarism. And those are things that get you in a whole heap of trouble, both at the university level and certainly in life. So please be what. But for me, I don't care. Anything that helps you get through this successfully works for me. I encourage it. I mean, if you didn't know, over an 8-month period, if you're in the military, they can change your job now. They changed, and my son's at a very high level. And all of a sudden, about 8 months ago, they said, "Oh, you are going to become our new expert on AI." I don't know anything about AI. It doesn't take long for an intelligent person to ramp up to speed. He goes around providing AI seminars and research and discussions on how to utilize it for the entire surface warfare of the United States Navy. He's that kind of high up. I'm patting him on the back. He's achieved a lot. Don't get me wrong. He's been in the Navy for over 20 years. Right? And all he does is, when I said I was exhausted from teaching on Monday from all you guys giving me a hard time, he had a very simple response. He goes, "Dad, retire." This is the kind of love and respect I get from my kids. Come on, you guys got to sell the house up here and move to Florida. No thanks. I have more important things to do. But do you get what I'm trying to tell everybody here? And frankly, it's typically the people who are here I don't have to tell this to. But for some of the people who are here are not doing as well as they want to do, please use this. It was like I was looking at my test. I'm being very honest with you, and I'm very appreciative. I really am. I went out of my way to be very thankful to the individual who was able to do this for us, and I continue to be. Enough said on that topic. On with the show. We all good? Great. So this stuff's important. From a standpoint of this is important today contextually, understanding how the nervous system works. Moreover, when we're going to be introducing neurotransmitters, and that's really the crux of this. So one of the things I sent you the stuff on the brain just now by mistake. What is that? 22B. I sent. Ignore that. That's pertinent to digestive, and I'm not teaching AP2, but I know you've had stuff from Dr. Robbins on the brain, so I'm going to have to pick and choose areas to cover, because it's going to make it. 11's got a lot in it, and 14's got a lot into it. And so once we finish this, sure, we'll finish this on Friday. Our time next week, and certainly for the week after we return from the break, we'll have to pick and choose what topics, and I'll adjust the final exam accordingly. On with the show. Well, that was hard to explain. Nervous system. Synapses are the junction. The neuromuscular junction is nothing more than a synapse. This is where a nerve and a gland or a muscle come together. And it's the same kind of thing. It's a fluid-filled space. It's small. We're going to look at the one we're studying. I'll talk about the other ones. The one that we have to pay attention to is the chemical synapse, in this case, in the nervous system, between the axon of the nerve that's transmitting and the dendrite, the receiving portion of the next nerve. And understand they all receive multiple signals, and typically they spit out multiple signals. So there's literally, right now with me talking to you or whatever else you're doing, there's millions of conversations going on in your head about what to write down, what to listen to, what I just said, whether to make all of that. Even the stuff that's peripheral to it, the voice that you heard over there, this over here. Why is it so freaking hot in the rooms today? You know, okay, that's what's going on in your brain. So, but we're going to look at nerve to nerve. Fancy term, the sender is called presynaptic. The receiver is postsynaptic. I tend to stay away from that language. It's confusing. When we get to the autonomic chapter, which is our last chapter, then you're going to hear terms like pre and post ganglionic. Those are kind of important. Okay, that one's where that really matters. So, and it's very, very true. Most function is both. When it's receiving signals from one and then sending those signals, modifying them, changing them, whatever, to the next one. So it's interesting how this occurs, and you'll see some of the complexity. This is what we're really, where those chemical signals, even though it doesn't demonstrate it here, you just see the contact point in blue from the axon and the receiving area, the dendrite, in that sort of golden or yellowish color. It's there. They're not the only, those are by far and away the most common. Sometimes you'll have transmission directly here to the body. That's probably the second most common. The soma is another name for the body, like somatic nervous system has to do with the body's nervous system. It really means the cell body. And it's almost like taking the signal and saying, oh, here, remember that signal you just got? Okay, well, it's not super important. Keep it on file, maybe we'll use it later. It's modifying some aspect of that signal. And now, even if you get it down here by that axon hillock, where one axon is contacting another, then it's probably saying something like, you need to send this has got to go FedEx overnight, or this one you can just do, throw it in the mailbox, we'll do snail mail and get it there, right? Do you have an idea? I'm trying to use some kind of analogy to bring the point home, that it's able to modify the signal. So the fact, and we're doing this all the time, it's part of our nervous system's development, which is unbelievable before birth, and particularly in the early years of childhood. And now you're in a learning environment here. I mean, you may or may not believe it, but a lot of that stuff you're assimilating, and it's going to be there hopefully when you need it. The trick is for a lot of folks here, and I've sort of morphed into test taking, is being able to study in such a way that you retain enough of it. The idea of a prerequisite class, like going into AP2, which thankfully you won't have to put up with me, is to have a passing knowledge, what we consider a passing knowledge of AP1 for any prerequisite course. If it's a math course, you have a passing knowledge, whatever that definition is. And the same thing, that's one of the prerequisites for the 50 or so of you who have foolishly decided to take me for micro. I just spoke with the other micro professor. Very nice guy, much more pleasant and user-friendly than I am, Dr. Maurer. You'll love him. Just saying. I think he's teaching next door. And this is what it looks like in real life. It's there. So there's an exceptionally large degree of complexity. So I'm not going to ask you any of these terms here, but the axon to the dendrite is what we're talking about. And really our focus point is going to be on the chemical synapse, because really the thrust of today is to start to introduce you to these chemicals. And the chemicals are really what this is all about. That's where we influence things. We'll talk a little bit about therapeutics. That's the kind of way when someone gets, I talked about calcium channel blockers. When someone gets a medication like Paxil or Prozac or Zoloft, and they're all very, very similar for eating disorders, obsessive compulsive disorders, ADD, mild depression, postpartum depression, but not psychosis. All these things that are comparatively common, basically they work through neurotransmitters. We modify them. As you'll see when I get to explain it, we modify the reuptake of those. More on that is coming. And so we'll look at the chemical. There are also electrical ones. They're more about once the signal is given, maybe giving it a degree of how rapidly or how slowly, some subtle modification. So we are really looking at this with neurotransmitters, the axon. This is all the neuromuscular junction revisited. That's why it reinforces what's going to be on the exam. In this case, this would be the nerve going to the neuromuscular junction. It's a synapse. The tip of the nerve has synaptic vesicles. In this case, they're filled with only acetylcholine. And at the neuromuscular junction, there's only one message to the muscle: fire. If we get enough of it over a significant period of time, the muscle will fire. Not so in the nervous system. Most nerves in the nervous system secrete more than one neurotransmitter based on a variety of factors, including certain frequencies from an electrical basis that are produced. Typically, those neurotransmitters work not necessarily similarly, but often oppositely. For instance, you have the nerve in your brain going down to the spinal cord that triggers the nerve that's going to the neuromuscular junction, can either tell that nerve going to the muscle to fire or don't fire. You can either excite it, make it more likely to fire, or inhibit it, make it less likely to fire. You all have been immunized for tetanus, okay, because we all are. So how does tetanus work? There's a neurotransmitter, basically, that the neurotransmitter that tells your muscle not to fire can't be released. So every time your muscle gets a signal, it means fire. And sooner or later, when it keeps telling it to fire, but nothing is going to relax it, it's going to go into spasm. What's tetanus? An unbreakable muscle spasm. That, oh, by the way, is fatal. Okay. We get to that in microbiology. Get any idea? Okay. That's why this stuff becomes important, because you build on it in subsequent courses. That's why, so next time if you're in a nursing major, say, why do I have another? I had a tetanus shot. Well, heck, you know, it wears off somewhere between 5 and 10 years. We can't take that chance. And they need to understand that the risks that are associated with it. Moving on. So axon terminal contains synaptic vesicles. The receptor receives a neurotransmitter, usually on the dendrite. It has the same kind of a cleft. It's a fluid-filled space, and yes, fluid can ease out of it. That's diffusion. And effectively, this is the rate-determining step. They'll use the same terminology that we do in chemistry. This is the slow part. That same son, when he's stationed in San Diego, and hopefully this time next year when he's stationed in Bahrain, in the middle of the Gulf, the Persian Gulf, you know, I'll talk to him in real time. It's going to take me a heck of a long time to fly there. That's the difference. It's why nerves are not going faster than that, although it seems ridiculously fast, that 100 or 200 meters a second. I mean, maybe that is 500 miles an hour. But nerves, but I don't have to wait 4 hours for me to say hello and him in California to say, hi, Dad. Right? We're talking in real time. This is the slow part of it. It may not seem slow to you, but in a human being sense, that tiny gap is still of some degree of significance. So we change it from electrical to chemical and then back to electrical. That's the time gap. So the synaptic cleft is important because it prevents nerve impulses from directly passing from one nerve to another. It's chemical, it's not electrical, all about release, diffusion, how well it binds, and it's all about unidirectional. That aforementioned tetanus, that gets into your system as a toxin, and it travels not in the direction the nerve is coming, but going backwards. We call it retrograde. Nerves are one direction. It fires in one direction. The signal is carried in one direction, but we recycle materials. There are roadways inside of the nerve that particular toxin accesses and uses it as a way to get back to the soma, the perikaryon, the nucleus or cell body. and take that over and say, we're not making any more of that other neurotransmitter. So these are well understood things, and that's why they're so important for you to understand. Not that people are going to ask you about the mechanism, but you can understand how serious it can be, because it affects literally every nerve going to every muscle, every skeletal muscle, certainly. I don't do that one. This is just a review of what happens. The action potential arrives from the axon of the presynaptic, the transmitting part, back to those good old voltage-gated calcium channels. They're still there. The same way the neuromuscular junction fire, all nerves fire. They come from outside the cell. That's extracellular calcium. I don't get a lot out of these illustrations, but that's what they use. Here are the synaptic vesicles. Here are those voltage-gated calcium channels. Here comes the signal, and it releases the neurotransmitter. You can see it's not as well organized or intricate, probably a better word, as the motor end plate, those infoldings, and all the surface area that's involved in it. It's totally different. It's a more simplistic, but we use it so much more frequently. Okay, and basically there's different kinds of channels. We're going to explore the chemical ones, of course. You're not going to have to know this name. This is that one. This is the chemical called, it's got a great name, synaptotagmin. You're not going to have to remember it. I always like to emphasize names, these bold-faced ones. There's a remarkable affection for certain names in standardized tests and licensure exams. This is one of those ones I take a little notebook and say, these are Stein's names that I might have to remember one day. Okay, but not for me on a test. Naming particular proteins not important. This is the one that's blocked by Botox. It doesn't allow you. Remember, you have the vesicles have a little protein. The membrane has a little protein. They snare, they interlock, and they pull it so it can spit out. This is the protein that it inhibits, the synaptotagmin. So that fusion results in exocytosis. But the whole idea, frequency, higher the impulse frequency, the more these open up and spit out, greater effect on the postsynaptic cell. You're going to see there's two things. You can either have things that are released very, very frequently, it'll lead to an action potential, or you can get a whole bunch released at once. And you're going to see some notes about this as we move on. So here's that stuff that's going on. Neurotransmitter diffuses, just like the muscle, binds, and again, chemically gated ion channel. Just what it does in the muscle. 3 sodiums come in, 2 potassiums goes out. The inside gets slightly more positive. If it's big enough and reaches threshold, the muscle cell will fire. In this case, the next nerve will fire. The big difference again is when it's acetylcholine, it only sends one message to that motor end plate, fire. So the protein changes shape. We don't call them end plate potentials in the nervous system. We call them graded potentials because depending on what the nerve, and there's thousands of nerves impacting it as simultaneously almost, they're not all saying fire. Some are saying don't fire. And the terminology we use is excitatory or inhibitory. More likely to fire, less likely to fire. And then the same rules apply, and acetylcholine is a perfect example again because it does all three. Most of them don't do all three. It can either diffuse away, because remember, it's not an enclosure, it's just a fluid-filled area. They will undergo some type of breakdown or degradation by enzymes. Very common. Acetylcholine does that. It breaks it up into acetic acid, which can diffuse away. We use that all over the place. And choline, which we suck back up by the astrocytes, called reuptake. And you can see it's milliseconds. It's not there. As long as the neurotransmitter is there, those potentials continue. And so it's taken away very, very quickly. Again, not necessarily by all three. Acetylcholine is a good example of all three that are there. And this causes the so-called synaptic delay, the rate-limiting step. So it's very quick, but this slows it down significantly. We don't notice it because how fast can I do this? How fast can I grab that? When something's falling, how fast can you catch it? Very, very quickly. We don't notice that delay realistically. And maybe, maybe that's that. Either way I look at it, it's that little button you have before, somebody just said something that just, and you want to respond, but you figure no. Maybe that delay is enough for us to control certain things. And obviously, some folks don't do it particularly well. Electrical synapses are there. They happen to work by gap junctions. It's very rapid. It can go in both directions. So think of how rapidly you look around and move your eyes. You're listening to me, but in the meantime, you put something out of the corner of your eye, you saw something over there, you heard something, all of that. Think of how rapidly we could do it almost without thinking, almost subconsciously. And then the hippocampus, an area involved in emotions and memory. I'm told Dr. Robbins commented on the hippocampus. That is an area particularly responsible for something called short-term memory. So what disease that older folks get affects specifically that area? People as they get older are subject to, yeah, what kind of dementia? What's that name we generically give to it? What my mom succumbed to. Begins with an A. Alzheimer's, thank you. Both, good, very good. Actually, its original name was SDAT, senile dementia of the Alzheimer's type. Specifically now with technology, we used to be able to only diagnose that post-mortem. Now, with the technology we have with scans, we can see areas that are deteriorating. They're typically in that area. The hippocampus, if you don't recall, is part of something called the limbic system. That's our emotional area. Okay, memories are tied to emotions. Part of it is the rage center. It's called the amygdala. That's part of it. We hear about people with road rage. There are disorders that are found there. Part of your sense of smell. Did they talk about the mammillary bodies? Is that ringing a bell? The olfactory nerve. Smells evoke really strong emotions. We can use smells that are linked to memory. It's the only cranial nerve that goes right deep into your brain. It goes into the cerebral area, called the old area called the insula. You remember that? Yes, no, maybe? See, all this is good stuff. See, I'm trying to put the pieces all together. That's why we use smelling salts. Okay? Because it's one of those things that smells they jog something called the reticular activating system. Kind of, oh, I'm awake, alert. I should have them here. That's why I try not to drone on in a monotone. Neurons are electrically coupled, joined by gap junctions that connect cytoplasm of adjacent neurons. Oh my. Now, if I did that, more of you would be sleeping than you are now. Opportunity to smile and laugh, commence smiling and laughing. Neurotransmitters cause graded potentials based on the amount and the time that they're there. Again, these are, terminology to be familiar with. Either they're excitatory, when they say postsynaptic, the next nerve is either going to be excited or inhibited. If it's excited enough, action potential. If it's inhibited enough, don't fire. And this is that fire, don't fire thing. That's all it is. Decision making, what you're going to do on an exam. Is this right or is this wrong? And a lot of things go through your mind. I remember this, I don't remember this. This seems plausible. That's basically what goes on there. The neurotransmitters bind open chemically gated channels, and these are the ones you're already familiar with. This is the one at the neuromuscular junction. 3 sodiums in, 2 potassiums out, and they are excitatory. It's only excitatory from acetylcholine. And yes, if there's a sufficient amount that stays there over a relatively brief period of time, guess what? We get an action potential, and that motor fiber, that muscle fiber will fire. So it's got to be of threshold strength, then it'll spread to that hillock or trigger zone, like you saw before, and then it'll get to those voltage-gated channels with sodium all over. Remember, it's not unidirectional in muscle, and boom, it'll be generated. Because there's 3 sodiums to every 2 potassiums, so there's more sodium inside. All stuff that's important to know for Monday. And they show it to you. That's an excitatory potential. Bring it closer to threshold. The ones, and this is how sedative hypnotics work. This is how sleeping pills work. There's also, and you'll become very acquainted, many of you will, with this. So tranquilizers work. Sleeping pills work. Alcohol works. They are inhibitory postsynaptic potentials. And most of the time, it is not these. It's not potassium opening. It's chloride. We call them chloride-gated channels. When they open, chloride ions flow in. They're negative. They make the inside more negative. They move it farther away from the threshold. Potassium going out would do the same thing. But this is, and you're going to hear a lot as we go forward about chloride-gated channels. They reduce the neuron's ability to produce the action potential as they move it farther away. You can see it there. So again, it can be chloride going in, but maybe less commonly potassium going out. And so, and we summate them. And depending whether they are over time, how rapidly they're being released, or that large amount over one particular space, that's called spatial. Not a big deal to me. But if there's a sufficient amount, either over time or over a sufficient area, that will likely bring it to threshold. You add them together. Most neurons receive thousands, notice the word thousands, excitatory, inhibitory, and only if the excitatories predominate will it generate an action potential. And that's what they talk about, temporal over time or spatial over a given. And again, I don't get a lot out of a lot of these illustrations. That's why I'm going through them. It's not something I'm going to ask you questions about. But here, it's quick signals, boom, fires. Over space, one big signal over a wide space, fires. And then we just add them together. So, this is the basis for some of the medicines we use. Here's the first one. Repeated use of a synapse increases the ability of the presynaptic cell to excite the post. The more we use it, okay, the more we're able to use it. Calcium concentration increases presynaptic terminal, causing release of more neurotransmitter, more PSBs, blah, 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 blah. So the calcium opens easily. And this is where, when you're thinking about how you learn, how we recall things, we use it. I say it to you so many times, and everybody else in all the classes. It's all about use it or lose it. All of us who were eligible, and unfortunately I was, based on the age criteria, they offered us a retirement package. When we had a new contract came in the last month. I said no. I sent it to my son, the one with the AI. He ran an elaborate AI program, and basically, financially, it was more or less not really great. It was very good. There's nothing wrong with it. It just wasn't great for us in our particular circumstance. The overriding factor. So he said, the first way he asked if you want to or not. I said, no. I want to be tortured looking to you guys continually. Yeah, I know. And I want to torture you guys continually. That'd be a better way to say it? I love interacting with you. So understand that's the first question in regard to these things. But my whole idea is everything is my concern. The minute I stop doing this, believe me, I can sit in front of the TV set all day and watch movies. What's up? Professional bull riding, darts competitions. I can find it. That's what use it or lose it is about. This is what happens. The most common set of medications that are used for mild or moderate psychiatric psychological problems are called SSRIs. It stands for selective serotonin, one of the neurotransmitters we're going to talk about, reuptake inhibitors. When you inhibit reuptake, if you block reuptake, it makes more of serotonin available at the nerve-to-nerve junction. The more, just like the more you use a nerve, the more calcium, the easier it is to use a nerve. This alters over time the receptor. This was the foundation of these whole category of medicines that many of you heard of, and more people will take them. Paxil, Prozac, Zoloft, they're all in this category. They are the different ones. Serotonin doesn't have one receptor. When you get into neurochemistry, there's all sorts of subclasses of receptors. And that's why there's so many different ones. They have different side effect packages. This has been the go-to for eating disorders, for obsessive compulsive disorders, mild depressive disorders, those kinds of things. Postpartum depression versus postpartum psychosis. It wouldn't be for psychosis. It's not for schizophrenia. So this has been, it was considered for a long time the go-to. Yeah, philosophically, there's a lot of things. Realistically, these medicines in and of themselves cannot cure something. It involves, there's lifestyle changes, there's counseling, most people speak. You need to have a lot of it. This is not my area of expertise. But this is a big, and the whole idea is we're basically tinkering with reuptake. Those acetylcholinesterase, that neuromuscular junction, those acetylcholinesterase inhibitors, it's great due to the myasthenia gravis. If you create it into a chemical weapon like nerve gas, you'll kill people. So that's something for mass destruction. Remember that. Going forward. That's something I think you may likely see on that exam. Just saying on Monday. Please. Okay. So that's what the idea of long-term preparation. This is how we learn. This is our nervous system developing as a lot of this stuff. So, and again, this is inhibition, and I don't get a lot. You can look at these if you want, have fun. We've seen all of these. Thank goodness we're at neurotransmitters. 2 minutes behind schedule. I'm trying. Oops. Neurotransmitters. There's about 10 that you're going to have to know. We classify them by their chemical structure. We'll look at that today a little bit and how they work. I'll talk about that one with serotonin in a minute. Chemical, and then the basic function is whether they're excitatory or inhibitory. And to be fair, some of them, it depends on the receptor. It can be, and serotonin is an example of that. Depending on the receptor, it can be excitatory or inhibitory. How those medicines work, we think, is you have somebody takes that, and over time it will alter the receptor, and that will provide whatever the benefit is. It's a good, understand, this is a slow process. For many of you who are going to run into this in fields which involve medicine, how should I say it? How many times has this happened to you? Maybe you carry Tylenol or ibuprofen in your purse or in your backpack. And somebody comes up to you and goes, I got a rotten headache. You got any Tylenol with you? And you go, sure. Let me have 2. And then 5 minutes later, you go to him, how's your headache? Oh, God, it feels so much better. That's called a placebo effect. Take a pill, feel better. And we rely on that a lot in medicine. Physiologically, it doesn't happen. It's going to take a minimum somewhere between 20 and 40 minutes for that to even start to absorb. And some of it, depending on what it is, if it has aspirin, it'll start to absorb even in your stomach. Ibuprofen, same way, a little bit faster. This is that. A doctor will prescribe this. They'll come back in 2 weeks. You want to make sure that they're not having an adverse response. They'll say, I never felt better. My whole life it doesn't work that fast. Those changes in the nerves don't happen in a week or 2 or 10 days. They take a month or 2 months to happen. But again, a lot of it, there's a lot of, when you're particularly in patient care, it's a lot of, if you're positive about something, people get encouraged. You don't want to lie to people, but I think you want to be encouraging. You have to be transparent, but some people need a little more encouragement. So be watchful of placebo effects. That's what they test when they do medication testing. It's called double blind, if you're not familiar with it. So they'll have people come in and they'll test a new pill. Half the people will get a pill that has the medicine. Half people will get a pill that's inert, a placebo. That's one blind. The experimental patient doesn't know. Whoever's asking, how do you feel after that? Did they give you an upset stomach? You have a headache, blurry vision? They have a whole laundry list of questions. They don't know who got which pill. The computer does. And so all that information in the computer is similar. In the old days, they had a third party submit all that information. That's called a double-blind study. It makes it more reliable. No bias. The most understood of all of these is the one you already know, acetylcholine. It's the only one in its class. It's a unique category. It's nearly always excitatory. You're going to see when we get into the autonomic nervous system, there's a couple of areas where it's not always excitatory. That's important. Okay, you're going to see that when we get to in the very last week of the class. Also used by many autonomic nervous system, and so we use it the neuromuscular conjunction, a lot in the brain, and in 3 of the 4 different contact points or synapses in the autonomic nervous system. Acetic acid and choline. Again, understanding the importance of this, very important, particularly for the test coming up. Serotonin is in this category of what are called biogenic amines. Way back, I talked about essential amino acids. A lot of what makes them essential and why we can't make them, but from our diet, is because basically they'll make chemicals like this. Typical amino acid, I hate to do chemistry, it gets you all upset. You don't have to know it, has an amine group on one side, R group up here. Remember that? So that's why I call it an amino acid. When you eat protein, you had a burger for lunch, chicken sandwich, a cheese, they're all protein. Those proteins, we bring them in, the only way we absorb them is amino acids. Typically what our body does is it deaminates them, it takes off the amine group. And if we're going to use it for assembly, it puts it back on and we have to make whatever proteins we make. A biogenic amine takes that amino acid and locks off the acid group. That's what makes it an amine. It's got one of those NH2 groups on it, whatever the chemical is. So we take the amino acid tyrosine, and it's super important because it's also thyroid hormone, which really governs how rapidly or how slowly your body works in general. It's made directly from tyrosine. Actually, it takes 2 tyrosines and some iodine, and voila, you have thyroid hormone. You learn about that in AP2. These are the catecholamines. They were originally thought to be the source of depression, not enough of them. They even came up with an inhibitor, like an acetylcholinesterase inhibitor, that inhibited the breakdown of these. That was the earliest type of agents that were used for depression. They didn't work very well and they had a lot of side effects. I had occasion to prescribe them they were helpful in chronic pain situations. One called diabetic peripheral neuropathy. That's where I had experience prescribing them. So I know a fair bit about them. Dopamine, this is called norepinephrine and epinephrine. We'll learn where they come from. Another name for these is noradrenaline, an older term, and epinephrine is adrenaline. Understand that going forward, we classify some of these receptors by the neurotransmitter. When we get to the autonomic nervous system, you're going to hear about adrenergic receptors because they're triggered by adrenaline, epinephrine, interchangeable names, or cholinergic receptors, they're triggered by acetylcholine. So that's why these names are important for me to give you some context going forward. So these are big players in your brain. Dopamine particularly plays a pivotal role in Parkinson's. I'll talk about that a little bit in the next chapter. Plays a pivotal role in addictive diseases. I'll talk about that. And it's made in, remember that substantia nigra in the midbrain, one of those anatomical landmarks there? It basically means black stuff. Okay, and that's where it is. That's where it comes from. Indolamines, and so one other little factor about it, they're typically excitatory. So if you eat foods that are rich in these, some people will be very susceptible. It'll accelerate their heart rate, increase their blood pressure. They'll begin to breathe rapidly. They'll get headaches. They'll be flushed. They respond, and there's certain foods that have a greater amount of these than not. The indolamines, indole is a chemical that these break down to, or they're in a very similar chemical structure. The serotonin and histamine, you all heard of histamine. Serotonin comes from tryptophan. Histamine comes from histidine. Tryptophan is something that many of you who are non-vegan will encounter 2 weeks from tomorrow. We find it in what? What's 2 weeks from tomorrow? And what are you having for dinner? We're having turkey. In my house, we're having both turkey and tofurkey. Vegan, you know. I love cooking vegan. So much tofu, and I even put in a little thyme. See, you gotta go for it. Tofam, we make that too. Merit beet juice. You make it red and all that. It's how they make those impossible, improbable, unusual, unlikely to succeed burgers. I'm sorry. I'm a carnivore. I'm sorry. Nevertheless, tryptophan what happens when you eat so much turkey at Thanksgiving? What do you do? What do you feel like after? Oh my God, am I tired? Of course, it may have something to do with the red wine or the white wine at dinner, but if we digress, right? But it makes you turkey. It makes you sleepy. That's what tryptophan makes you sleepy. Histamine. Histamine is contained in white blood cells called mast cells. They accumulate, and when they explode open, which they do, particularly with allergens, if they're clustered in your respiratory area, they can lead to asthma or anaphylactic shock. In your gut, we have histamine receptors. You'll learn about that in AP2. These other things you'll learn about in immunology chapter. Give me a minute. Almost done. So understand that's where antihistamines work in your brain as well. All of those things. Widely used in the brain, play a role in emotional behaviors, biological clock. So it's important to understand. Now, serotonin and histamine, it depends on which receptors there are, what they do in the brain. My daughter was taking one of those, a more advanced agent for her, she had bipolar 2. She developed a really bad complication when she was in March of 2001 called tardive dyskinesia. All of a sudden, I'm getting a call. My God, she can't, her tongue's sticking out. She can't get it back in, and her neck's stuck like this. You would think horrible. And, you know, she didn't have a fever, so you weren't thinking about meningitis, which is one of the differential. We had to run over to the hospital, St. Margaret's over in Aspinwall here. And she ended up admitted for 3 days. They had to give her intravenous Benadryl. Benadryl, which you can buy over the counter in capsules for mild sleep, mild cough, give it to babies, that type of stuff. She was getting it intravenously, and it was the only thing that allowed that to release. And it's very, very dangerous to get that intravenously. So I understand they play a significant, so these both have very, it depends on the receptors that were being triggered. That medicine that she was taking made a lot of these available, and one of them was serotonin. That's one of the complications because they're tied to movement disorders like Parkinson's. That's a big problem with all of these medicines that are there. And so we will look at all of those. And again, imbalances in these, and I think these days we're really more focused on serotonin than almost any place else. And I'll have something to say to you about that. You'll get more of that as you go on. And the last category that I'm not going to complete, I'm just going to make you aware of, even amino acids by themselves. Put that acid group back on. Glutamic acid and aspartic acid are amino acids. When they're in water, or they're in your bloodstream, in your plasma, in tissue fluid, they have a new name, glutamate and aspartate. They're the same thing. That just means they're dissolved in water. They are typically excitatory. Glycine, and this one we always just use the term GABA, which is the way you should learn it. Gamma aminobutyric acid, they are consistently inhibitory. So you have excitatory and you have inhibitory transmitters that are there. They are really, most of these work by opening those chloride gated channels. So it's very, very interesting. And these, again, and that's why we have such a myriad of uses that's why we have to have all those proteins. We can make some of this stuff from other proteins, those essential amino acids, these are all vital substances we have to have. So if we don't get that in, so if you're not eating that tofu, or something like that, particularly for vegans and less so For vegetarians, it creates problems that are there. Thank you very much. We'll meet again on Friday. Till then, bye-bye.